Hello, everyone, and welcome to Empower Your Bioanalytical Lab through Software Advancements. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please go to thermofisher.com. We do encourage you today to participate by submitting any questions you may have during the presentation. To do so, all you need to do is put them in the Ask a Question box and click Send. We'll answer as many questions as we have time for after the presentation. You can also submit any technical issues as well if you're having any trouble seeing or hearing the presentation. I'd now like to welcome our speaker, DJ David Joyce, Director of Software Product Management, Digital Science and Automation Solution from Thermo Fisher Scientific. For a complete biography on our speaker, you can click the biography window on the left of your screen. DJ, you can now begin your presentation.
Thanks very much, Matt, and welcome to the webinar. Today I'm going to take you through a set of solutions that we have applied to the bioanalytical space as Thermo Fisher. Primarily around software, but also talking more about how we interact more closely with lab automation as well. As we said at the top of the webinar, I'm part of the digital science and lab automation team, and most recently we've combined those two aspects of Thermo Fisher in order to give much better solutions in STEP space. So what I'd like to do this morning is take you through an introduction to our company. We'll talk about the challenges that we believe you're facing in this bioanalytical space. And then we'll kind of show you the path to the promised land, if you like. We're going to show you how we can better coordinate your data, tie that up potentially with lab automation and instrumentation, so that you can actually get much better throughput within your labs, much better quality in your bioanalysis, and ultimately, get your solutions into market much more rapidly. So we'll start with an identification of the challenges, then show how those challenges can be addressed using LIMS and automation tools. And then finally, we'll finish up and talk about some of the work that we've done around automation and digital transformation. And if we have time at the end, we'll have some questions. So to get us started, I'll introduce the company a little. I think many of you probably know Thermo Fisher from your working life, but I think everybody's got a different picture of it. The elephant has many different ends, and some people think of it as the supplier of their consumables. Others reference it through the Fisher catalog. Still others will think of it as an instrumentation company. But we're a multifaceted company, and I think one thing that would probably surprise many of you is that we do spend an awful lot of R&D investment. You can see $1.5 billion in R&D spent every year. About 250,000 of that is spent in software R&D. So many of our instruments, they are shifted with, they come with software to support the specific workflows that they carry out. But also our business, digital science and lab automation, is pure play software. We sell enterprise software that facilitates the work within many people's labs across the world. So we've got about 10,000 customer-facing representatives across the organization. Some of our customer-facing folks have been working in bioanalysis specifically for 25 or 30 years. Often they've come from a customer or come from a CRO or a sponsor and have that sort of that background. We then sort of parlay that into knowledge of the system, so they can talk your language when you speak to them. I'll move on to the next slide. We're driven by a mission. We aim to make the world healthier, cleaner, and safer. And we take pride in our mission. It sounds a little bit trite, I know, but personally, I have been lucky enough to work on some amazing projects over my 25 years in Thermo, including things like the Amplitude project, where we took our lab automation and our software, built a deck which we called Amplitude, and we built about 100 of those decks. Those decks could do 8,000 samples a day with only 3 people supervising them. They run 24/7, and over the course of the COVID pandemic, we tested about 56 million samples on those decks that we built across, and that was just our business unit. Obviously, we had a lot of input into the actual making of the tests as well. Other things that sort of I feel very proud that they're working on are things like the enhancement of cell therapy and CAR T work, whereby we build the instruments and some of the reagents and software to take cancer cells, modify them, and re-inject them into individuals, and that is a revolutionary sort of approach to cancer care that we are trying to bring from being hundreds of thousands of dollars per injection to something that is more manageable and more scalable. So we really do take a pride in that mission. And as I mentioned earlier on, we're quite a diverse company. So some of you might know us from the sort of early phases of clinical research, where we provide the reagents and the kits and the capabilities for carrying out analysis on the benchtop. But that then sort of moves into our sales arm, if you like, thermofisher.com and Fisher Sci. They give you access to these things and delivery in a timely manner. And then our part of the organization, digital science and lab automation, kind of works with all of these various areas within the company, especially some of our services groups. So if you look at the bottom there, PPD is our CRO, and we work very closely with those guys in some cases to hone our techniques and to get feedback from our internal customers, if you like, to enhance our software capabilities. And then ultimately, when you come up with your molecule, you've taken it through that trial process. We can also take care of creating and delivering that with our Patheon CDMO team. I think another thing that I absolutely love about being in this organization and the diversity of the organization is, I get to talk to some of the smartest people in the world on particular topics. If I pick up the phone and I want to know something about Orbitrap mass spec, Alex Makarov will take my call, and that's sort of a culture within the organization. People do reach out across the business and sort of use that tacit knowledge within the organization to help others within the teams. So, where's this presentation going? Where do we really want to be? Like, where's Nirvana there, the Holy Land? What we'd like to see in our laboratory is that our data and automation are working in harmony, and we're reducing the drudgery and reducing the potential for error within our workspace. So obviously, by bringing in digital automation, you can ensure that you make fewer mistakes in transcription of data from instruments to your system of record. You can remove some of the tedious calculations that you may have to do. Even with the most diligent work, you're going to make 1% to 2% of errors even in transcription when you put maths or any sort of stress in the situation. And obviously there's no stress in the laboratory, but if there's any maths or stress in it, then your error rate goes up to something more like 5%. So from a data automation point of view, that's why we need to have systems of record that actually do some of that work for you and ensure that you don't drop the ball yourself, and sort of has checks and balances to ensure that you've done the right thing. That then moves on to being able to deliver that at scale. And that's where the partnership we have with our lab automation brethren really comes into form. We'll talk at the end of the presentation about some excellent work that has been done by the automation team around phase 1 bioanalysis to really almost squash the error rates and double the throughput of samples within their particular customer's space. And obviously, we want to ensure that we we keep ourselves within compliance. So a large part of what we're doing is following those regulations and essentially ensuring that we're dotting the I's, crossing the T's, and delivering the final tables that are required by the regulatory bodies. So I'll go through some of the challenges. This is sort of feedback we've had from our own customers. Hopefully, a lot of this stuff will be self-evident to yourselves on this call, about areas that are challenging within the space. So we'll deep dive into each one of these titles, tease out some of the challenges that we face, and then we'll move on and talk to the solutions that we can help to provide in these areas. So, challenge number one: harmonizing your laboratory practices. It's a very specific process that you need to go through in order to create a method, validate it, and get it into the execution space. There's a number of different steps that you need to go through, and each one of them has to be carried out correctly before you go to the next. And then you need to be able to carry those things out, those assays out in a repeatable and consistent manner. So that's not easy. So having a tool that allows you to carry out those steps, capture all of the data from those steps, and have a record of how you've come about making the assay is essential. Applying the criteria across many assays. So you're going to be carrying out tens, if not hundreds, if not thousands of assays over the lifetime of a study. How do you ensure that those criteria that you've set up are always delivered in the manner that is expected? Having one place to go to access that data, to bring all of that stuff together, rather than multiple spreadsheets or paper systems, really is key, especially when you've got a long-running study that is maybe got thousands of analytical runs, hundreds of subjects who are constantly on the go, moving, coming in and out of that. Many sites you'll be transferring data from and to. That gets tough. So having something that is dedicated to doing that work and ensuring that your paper trail is managed is essential. Just sharing data. So this is all about the data. We don't do this for exercise or for fun. This is about delivering the correct tables to the regulatory bodies, so we can move our drug along in its pathway to the market. In order to do that, we need to share data with multiple different areas. So we need to share data with the regulatory agencies ultimately, but also there are handoffs of data between the sponsor and the CRO. Now, if you're doing that with paper, if you're doing that with spreadsheets, there's a chance that you can drop the ball or misinterpret data going backwards and forwards. You have to really craft a technique for sharing that data with your CRO and that tech transfer aspect, de novo each time. If you use a more standardized system, you're both speaking the same language. If you can send an archive of a study from your sponsor to the CRO, that data is transferred without any chance of mistake or misstep. Something that still blows my mind. I've been in this industry for 20 years. I took a little sabbatical and went into the cloud industry a few years ago. When I came back, I was still almost incredulous to see that we are still wrestling with sample manifests coming from study sites, sometimes from the central lab, that are inconsistent. People don't fill in every box, and sometimes you do get badly described samples. My brother's a CRA, a monitor on studies for a CRO, and he's still pulling his hair out about how we get this data from A to B. So our systems do have capabilities for kind of reconciling some of that data and making it easier to ensure consistency when you get these incorrectly put together manifests of samples. And then it's time and resource intensive to build these the reports that we need to build. So that seamless flow of data will ultimately save you time and ensure that you have a consistent outcome. There are also bottlenecks. So, when studies get large, some of the studies that we see are multi-year, long-running studies. People will be taking in, samples over an extended period. You need to ensure that the data is built up in a consistent manner, and you're not getting, after 2 years, you're not doing something differently than you were doing at the start of the process. So um, there's an aspect of drudgery, if you like, as well in these studies, building these studies. How do you ensure that your samples, your standards are being maintained? Whose job is it to ensure that things are within their required life cycle? They haven't expired. Um, how do you consistently apply the rules for doing reassays and incurred sample reanalysis? Again, this is something that is, maths is involved and a little bit of stress, perhaps. That there's a chance there that you can make a misstep, unless something is actually has your back and is actually walking you through these processes and ensures that you don't use a standard that's been has expired, or that you do the correct number of incurred sample reanalyses even. And then, should you be spending all of your life formatting data in spreadsheets and in Word documents, or can that be done for you, and you can be doing much more useful scientific work? And then finally, you want to maintain compliance. Back to that sort of drudgery aspect again. Things like tracking free store events. Do you do that on a piece of paper? How do you then map that into your final reports, or is that done for you? Every time something comes out of a freezer, it's moved. Is that recorded? Can that be done in an automatic manner such that you don't misstep there? When you have a paper-based process, it's up to you to do the right thing as far as applying those rules that we've described, and also, applying the rules necessary in your assays for reassays when something fails, or ensuring that sample is realiquoted and done again. So, that's a tough thing to do, and it's day in, day out. You need to be consistent, and then ultimately you need to be able to prove that you've done what you said you've done. So you need to have an audit trail of your actions. If that's on paper, that's an awful lot of signatures. It's an awful lot of paper to manage and to work through. So in an electronic system, you're much more likely to be able to keep that consistency. Also, withstand those audits that come through from the governing bodies, or even from your sponsors, just to be able to prove that you've done what you said you were doing. So I think probably those are kind of well-recognized issues within the space. How can we help to ameliorate those issues? So I'm going to talk about LIMS software here, laboratory information management systems, and how a dedicated bioanalytical LIMS can help you get over some of these challenges. So, we've talked about the challenges without LIMS, regulatory practices and guidance documents. How do you maintain that you're carrying out the right number of incurred sample reanalyses, for instance? What does that actually mean? If you're not using a system that is designed specifically for this type of process, then that's on you to work out exactly what protocol you're going to follow. We've talked about the free store cycle. We've talked about streamlining and making the processes more consistent. How do you hand off your data between an instrument and the final results? Does your LIMS system capture that data? Does your manual system capture that data? I would say not. So we can promote compliance. You can implement assays for not just the PK analysis within our LIMS system, but also the anti-drug antibody immunogenicity work that you must do these days for large molecules. You get to track the life cycle, not just of the sample, but the complete study. In a bioanalytical LIMS like Watson, you will essentially have a full picture of the study, its subjects, their movements, also all of the sample transitions and transfers that go on between study sites and the lab, and you have a like a 360 view of the processes going on within your laboratory. So I mentioned the name Watson, Thermo Scientific Watson LIMS software. It's been around for almost, I would say, 25, 30 years. I've been working with it for 24 years. And it has evolved over that time to be, the world leader, I guess, in the field as far as being a specifically designed LIMS for bioanalysis. All the processes it carries out are driven by the regulations, and given its sort of breadth across sponsors and CROs, it eases the transition of data from a sponsor site to the CRO site. It also it has full audit trail of everything that you carry out, from the study design, the assay method validation, to the execution of that, and then ultimately the digestion of that data, pulling together the PK reports and the other ancillary reports that you need to submit to the governing bodies, and then your samples are fully tracked as well. There's no chance of losing anything, even not all the way through from that registration of the sample initially to the creation of it, to its movement through the various sites, to ultimately to the disposal of that sample. So as I mentioned just then, that we have a complete assay system for PK as well as immunogenicity. As you can see here, we walked through the steps of the tiered approach, from screening, confirmation, and then titration of your large molecule experiments. And you can then also use this tool to calculate your assay cut points, ready to carry out your tighter work. All of that's in one place. So if somebody comes and challenges you on a particular set of values for a particular individual in a particular study, and you can show that you have done the due diligence to actually carry out the assay development correctly, and you can tie that, it's all in the one audit trail. You can tie that back to from the initial design all the way through to the result value that you give to the FDA. As far as reporting is concerned, we are driven by the regulations, and we have defined the reports that are required. Those things are generated within the LIMS system, multiple different tables, and the LIMS system has a document management system. So essentially, each one of those reports you create, you can actually push into our document management system, and again, you've got the audit trail of everything that you've done there. You can see where the data came from. You can then map it to the tables and back and forth. And then you can quickly apply your Word templates. If you have a specific template you wish to use, you can apply that template to all of the tables and aggregate them into a single document. Again, all within the same piece of software. So I'm repeating myself a little, but the results, the reports, and the original data, and all of the sample tracking can be tied together in the one system. So you have a one-stop shop to prove that you've done the right thing and you've provided the right data to build your reports. You can do your PK there and do your QA and QC reviews as well. And once you have pulled together all of the data for your study, obviously you want to then audit that. You want to make sure you get second eyes on it. You go through and do the correct sign-offs on the data. You can lock that study and make it read-only. You can then allow the audit of that data to go through and check things out before you generate those final reports. And ultimately, the you have the ability then, once the study is completely finished, to archive that complete study in a method that will allow you to retrieve it fairly easily and bring it back to bring it back into the front of house. Sorry, the archiving capability is necessary when you look at a big CRO and they've been doing thousands of studies over an extended period. You don't want all of those studies to be live and online in your system, because that is actually it sort of taxes the capabilities of the computer system if it's all live and active. So the ability to be able to archive this data off from the system allows the system to continue to maintain speed and agility, rather than being sort of bogged down by a lot of data that's kind of in the live database. So that would be one reason that you might want to archive studies off from a system. What you do need to know, though, is that you can bring back that data at any time and allow your sponsors or your governing body to look at that. You also might archive a study such that you can actually pass that data consistently to a CRO from a sponsor. So we mentioned this; it was one of the challenges at the top of the call, whereby it's sometimes inconsistent the way that sort of the tech transfer between a sponsor and a CRO is carried out. If you do it within Watson, both the sponsor and the CRO can have a copy of Watson. You can define your study, define your assays in-house as a sponsor, and then create an archive and transfer that digital archive to the CRO, where they can de-archive the study, and you have an exact transcript, an exact copy of the work that you've done in-house, available to be used directly by the CRO. No chance of changing the reassay decision tree, or no chance of transcription error as you transfer that data from sponsor to the CRO. So I'm laboring the point, but the fact that things are done in a digital manner and in a single place, this is a single file, ensures that you both have the same view of the study that you're carrying out. And another burden that we take on, as opposed to putting that burden upon the customer, is that we work to ensure that the software has all of the current industry best practices built in. So if ICH changes their processes and expects certain new calculations to be carried out as part of the study workup, it's not on you to go and work that out in Excel and build that type of capability yourselves. We actively maintain our software. We add new versions of that software on a regular basis to try and address these needs of the regulatory bodies. That way, everybody is doing it consistently, and it's not something that you need to keep on top of. Obviously, you need to know about these things, but it's not something that you then need to codify into your software or into your practices in order to keep within the bounds of regulation. And some of the issues that you see, I mean, that when you do start to use any type of computer system within this environment, it needs to be validated. The process is somewhat onerous. Things have changed probably in the last 20 years, whereby there's a little bit more of a risk-based approach to validation is being carried out, whereby you emphasize the testing of the parts of the system that will have potential influence on the data quality. You emphasize that more over than, say, the printer drivers or some of the user experience type of things. But that being said, if you use something like the Watson LIMS software, Watson is very codified. You have a selection. You have the rules already in the system, if you follow the steps that are defined within Watson, you will be doing the right thing by the authorities. The chance or the ability to deviate from that is limited. Now you can decide your number of decimal places you're going to work to, the reporting formats you're going to work to. There is certain flexibility in areas that are not critical, but certain aspects are coded into rules are coded into Watson such that you don't make mistakes. Your software is not going to be incorrectly calculating values that you need to calculate. The benefit, or one of the main benefits of doing that, is that instead of customizing your software, so if you were to use a generic LIMS package, so a package that is customized from the ground up by the user and the vendor's team for a specific purpose, if you are doing that from a sort of generalized system, the validation burden is much higher, and that validation burden is on you. The differentiating feature, I would say, for Watson LIMS is that this is all codified in the system, and it's much easier to test. We have a suite of validation scripts that you will run. So you have the installation qualification document where you show it's been installed correctly. And then there's a very comprehensive operational qualification. That operational qualification is a process where you go through each one of the functions of the system and ensure that it complies to what it did when it left the factory, if you like. So you're confirming that the system does what it's supposed to do, and does it in a consistent manner. That documentation would have to be written from scratch for a custom LIMS system, because it will be different from one installation to the next installation to the next installation. Within Watson, that's a package of papers that you can actually work through, and it's something you can buy from us, I suppose, as opposed to having to do that from scratch. It really saves time as far as your validation efforts are concerned. Going forward, we've started to carry out automated testing on our products in-house, and we're looking to actually provide that automated testing capability for OQ scripts to customers in the near future, such that we can really compress the time it takes to actually carry out this on-site testing and OQ testing. Typically that can be a matter of months to actually carry out all of the tests on the detailed functionality of a system. It could be tens of months if you're doing it on a custom system. We hope to be able to compress that into weeks, if not shorter time frames, at least for the OQ testing. Performance qualification, that is kind of the final user acceptance testing and showing that it does what you require in your own context. That's something that still is going to be some work for yourselves. Only you can sort of define that it's doing what you need it to do. But that OQ can be sort of reduced significantly and reduce your time to value by many months. So now moving on to the exciting stuff. I've just come back from the SLAS conference in San Diego this week, the Society for Lab Automation and Screening. And I saw some fantastic examples of connection of the digital and the physical world. As I said at the top of the call, I'm very happy to be working now very closely with our automation team. In fact, we merged the software and automation team just before Christmas, because there are such synergies when we actually start to work together. So for those of you who don't know about our lab automation capabilities, that's a business unit that's been with us for nearly 20 years now. They have best-in-class hardware, so they make things like the Spinnaker and the Orbiter, the various plate handling robots, lots of plate movement sort of systems to take your your plates from A to B to the liquid handling systems. We also make cytomats, so automated incubators and shaker incubators. And all of that is coordinated using workflows and scheduling software. In our case, it's called Momentum. And that allows you to integrate not just Thermo Fisher instruments, but third-party instruments. I think Momentum has over 300 different instruments it can control. Integrate those into decks, which will automatically or semi-automatically carry out the activities in the laboratory that you would normally carry out manually. We had great success, as I said earlier in the presentation, with a solution that we put together for COVID, the Amplitude solution. And that's kind of was a precursor to us working together with the lab automation team to better integrate and coordinate the digital workflow as well as the physical workflow within the laboratory. So we have the next slide is a case study that we carried out with our customers, Moderna. I had the pleasure of meeting Juan yesterday, and he was taking me through what they had done. But what you're seeing here, and it's a little bit of an eye chart, I apologize, is a project that was carried out probably in 2023 and presented in 2024, where Moderna were carrying out phase 1 clinical testing at high volume testing for flu vaccines, and they wanted to ensure, speed up that process. There's a lot of samples in a vaccine test that you need to get through, and they wanted to reduce the failure rate, increase the throughput, and reduce the amount of human effort that is required to get to their goals, and do that in a consistent manner. I think the other thing they were looking at as well is how do you scale this type of process. So they wanted to prove that, first of all, it was possible, it was feasible, and it was valuable. But they also wanted really to think about this approach of whether you can sort of, as I say, on sort of your shampoo, rinse and repeat. It's very difficult to set up a new lab which will do the same testing and then do the tech transfer to it, and then carry out the testing consistently. Doing that with humans in the equation is a difficult task, and it's something that we do all the time, but it's difficult and expensive. The hypothesis that we're testing is, well, can we automate this such that if we do want to scale up the frequency of the trials, we can make another one of these systems and validate it. And validating a system like this is a much shorter process in time than actually building a new lab and the various training that will be necessary and equipping that will be necessary to do that. So they were kind of testing the hypothesis that they could scale using automation. So the highlights were that they were aiming at a 2 times increase in throughput of samples, and a reduction of failure rate by 5%, sorry, 5-fold. And they hit that target. So you can see there that the throughput was doubled as required. So it went from 5,000 samples in the period to 10,000 samples in that month period. Very few retests was also one of the outcomes here as well. The consistency in the pipetting and the incubation and the various different steps given by the robot automation system really sort of removes a lot of the potential for error. And you can see that the fail rate there dropped from 6% when it was done manually to 0.37% over that period. And each one of the individuals involved, well, of the 4 people who were involved as opposed to the 9 who were in the lab before, were 3.8 times more effective through carrying out tests. So it was a clear win for that approach, and there's some ancillary sort of study work that we've done that kind of looks at how we could even go faster and do this even better. I'm coming towards a close here, but I'd like to mention some of the work that we've done alongside our colleagues at PPD. So PPD is a CRO that is part of Thermo Fisher, and we work very closely with that team. I mentioned them in this automation sort of sense of the word because one of the things that they are also trialing is a similar hypothesis that Moderna were talking about. This idea that we can work with a customer to build out their automation system. They have their digital capabilities, as far as the data management is concerned, and then do that in potentially on their premises, and then PPD can then take over the automation system from the customer, such that they can then scale. So if the customer wants to scale from 100,000 to 300,000 samples a year or what have you, PPD can work with our automation group and ourselves to actually build one more or more of these robotic systems to carry out that sort of testing at scale. And then obviously PPD will run those things, and they're past masters at doing this. During the COVID era, they had a huge push towards automated liquid handling and automation of the activities needed for bioanalysis, obviously mainly for the testing of COVID tests, out of necessity. You wanted as few people in the lab as possible and as consistent a result as possible because people's lives are on the line. So we are in a very valuable position in that we're able to work with those guys, learn from what they have learned, and bring that type of solution to the broader market, and share it with the industry. So that's our aim going forward, and I'm really looking forward to the next few years working alongside my automation and CRO partners to improve our software even more to try and facilitate this unique use case. So with that, I'll round it up, and we'll take some questions in the chat, I believe. But the key takeaways, I would say, is there is a real tangible benefit to optimizing your operations, and this will give you a competitive edge using a LIMS system. The consistency it brings, and the simplification of the process, and the better ability to communicate between sponsors and your sponsors and the CROs, ensures that you're doing the right thing consistently and getting your drugs to market much more quickly. The exciting piece that I keep talking about, this innovation between physical and digital automation, I think is going to be much more apparent in the next few years, and I feel that we're going to be on the vanguard of that with our automation team and PPD. And if you want to talk about doing that for yourselves, come and speak to us. We aim to be the world leader in serving science. We have the breadth and the knowledge to provide the physical and the digital automation that you need, and we have the individuals, the people who are passionate about this, who will take you through that process and make sure it's a painless transition to the brave new world. And with that, I'll say thanks, and we'll take a few questions.
All right, excellent. Thank you for that presentation. We'll now start going through the live Q&A portion. So if you do have any questions you haven't sent in yet, go ahead and put those in the Q&A box and click send. So let's start with this one here. In relation to sample chain of custody, you did mention capabilities around barcoding for sample management. Can you expand on these features? And does Watson require additional software to support that label creation?
Yeah, thanks for that. I can remember when barcoding was seen as sort of strange and high-tech technology, and it's been around for quite a while, but people seem to be scared of it. It's been something that's been a fixture of Watson for maybe 20 years. We have a very specific piece of software built into the package to design and make your labels and your barcodes, so you can do that in a very flexible manner. but also we cope with the fact that you don't always get to specify which label you're going to use. So Watson also has a capability around being able to use external barcodes and map them to the internal barcodes that you might want to stick on the samples yourself. So, we've got a digital method to reconcile between, maybe a third party's barcode and our own, and we have a full capability around how those barcodes can be generated within the Watson LIMS itself. Thanks for the question.
Can you tell us more about the automated validation plans?
Okay, it's a topic I'm quite fond of at the moment. Having spent my earlier career validating Watson LIMS systems and going to places such as Fargo and various other out of the way places, and sitting for weeks potentially, going through the process of checking every function of the LIMS system to ensure that it was functioning correctly. That was intensely, time and labor intensive, and the ability now to take they call it a robot process automation tool, RPA tool, to actually do the work that I used to do with a pen and paper and my mouse and keyboard, is a game changer as far as I'm concerned. The initial process we went through was we built out the operational qualification tests in a tool that actually automates the mouse clicks and the keyboard presses, and then takes screenshots of everything that it does, so you can see the steps it's carried out, compares those screenshots and the values that come out of the system to the nominal ones, the ones that we're aiming for, and then provides a report that you can query instead of like a paper report that you can't search. This is a report that you can search through, find any deviations, quickly correct them, and move on. And to run those tests, instead of it taking many weeks, it can be a matter of hours. Let's call it days, because you need to do some quality checking. But I'm really excited to see that capability coming to the system and removing some of the absolute drudgery that is necessary in that testing space. So yeah, watch this space.
Okay. So it looks like we've got time for one more, and any other questions we don't get to today will be followed up with after the fact. For CDISC support, will Thermo Fisher continue to introduce new CDSIC domains
Yes.
to support future implementation guides?
Yes. So I mean, it's great that that CDISC has sort of been adopted by the industry. Again, I'm going back a long way, but it feels like recent memory for me, 15 years ago, something like that, where everything was sent as Word documents, and the data would be transcribed by the regulatory body from these documents, and they would then second guess and check the values. CDISC was a real revolution back in the day. It's an XML format where the data that we capture and the data tables are actually represented in a way that a computer can read, and the ability to do that means that there's no chance of transcription error of taking that data from the tables in a document into the calculation programs that would be used by the FDA. So it's been a revolution to do that. We always keep up with the regulations. I mean, when the regulations change or the formats change, we have a little bit of a lag, because we've got to work out how do we develop the software such that we can support that. But it's our commitment that we will get to bring those new structures into the software in a timely manner, such that. So the answer is yes. We're going to support the new formats of CDISC as they come along, and I'm encouraged by the fact that the FDA is moving in that direction, because it leads to much more consistency of data transfer.
Right. I'd like to thank you again, DJ, for your time and your presentation today. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's webcast. Before we go, I'd also like to thank the audience for joining us and for the questions. Any questions we didn't have time for, and any questions that do come in during the on-demand period will be addressed via the contact information you provided when you registered. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available. We do encourage you to share that email with any colleagues who could have missed today's event. And until next time, thank you very much and have a good day.